that's the beginning of, you know what I mean? The last thing. So, what? Just do it again? She just said it. So,
Reinforcement's
anyway.
always
What?
good. Reinforcement's always good.
Okay. Hang on a second. Alright, so, in 18.2, we learned that, bacteria coordinately controlled genes are often clustered into a, what? Does anyone know? Maggie?
Chromatograph?
No. They're clustered into, any
What does it start
guesses? Sean? I'm sorry, Dr. Ahmed, as you, so I just called him.
What does it start with?
It starts with an O.
Operon.
Yeah. And an operon is, no one knows? Al?
Everybody knows that.
Okay, again, an operon is a unit of genetic function found in bacteria, and it consists of a promoter, an operator, and a coordinatedly regulated cluster of genes. And they all like perform a common function in like a certain pathway or something like that. And an operon is regulated by a single, I just said it in the definition. Yes. I mean, no, promoter. And is, the next blank, it's something, it's like a verb into a single mRNA molecule. This has to occur for it to become an mRNA molecule. Sam? Yeah, transcribed. Okay. All right. We kind of just went over this. Genes are expressed together and encoded in proteins and are produced at the same time. Operons that work in this manner have not been found in eukaryotic cells, but there are some exceptions to this. In bacterial gene operons, each gene in such a cluster has its own promoter and is individually transcribed. In other cases, several related genes share a promoter and are transcribed into a single pre-mRNA. And unlike bacteria, RNA transcript is processed into separate mRNAs. Okay, co-expressed eukaryotic genes are more common, and they include genes coding for enzymes and on the metabolic pathway, and they're scattered across the chromosome. In these cases, coordinated genes expressed most likely depend on the association of the combination of control elements with every gene of a dispersed group. And the presence of these elements can be compared to the raised flags of the mailboxes. Copies of activators that recognize the control elements bind to them, promoting simultaneous transcription of genes no matter where they are in the genome. Coordinate control of dispersed genes in the eukaryotic cell often occurs in response to signals outside of the cell, and an example of this can be seen in a steroid hormone. A steroid hormone enters a cell and binds to a specific intracellular receptor protein, forming a hormone receptor complex that receives transcriptions of an activator. And every particular gene whose transcription is stimulated by a particular steroid hormone, regardless of its chromosomal location, has a control element recognized by that hormone complex. And this is how estrogen activates a group of genes that stimulate cell division in the uterine cells, preparing the uterus for pregnancy. Many signal molecules such as non-steroid hormones and growth factors bind to receptors on the cell surface and never actually enter the cell. These molecules can control gene expression indirectly, and they do this by signaling transduction pathways that lead to the activation of particular transcription activators or repressors. The principle of coordinate regulation is the same case as in steroid hormones. Systems for conducting gene regulation probably arose early in evolutionary history and evolved by the duplication and distribution of control elements within the genome. And genes with the same control elements are activated by the same chemical signals. Transcription alone does not constitute for gene expression. The expression of a protein coding is ultimately measured by the function of proteins a cell makes, and much happens between the synthesis of RNA transcript and the activity of a protein cell. Researchers are discovering more and more regulatory mechanisms that operate at various stages after transcription, and these mechanisms allow a cell to fine-tune gene expression rapidly in response to the environmental changes without having to change their transcription patterns. RNA processing in the nucleus and exporting of mature RNA to the cytoplasm provides several opportunities for regulating gene expression that are not available in prokaryotes, and an example of this is alternative RNA splicing. And then there's a picture of this here. And so basically the primary transcript of this gene can be spliced more than one way, generating different mRNA molecules. And we notice that one mRNA molecule has ended up with an exon 3, and that's the green one right here and here. And the other with an exon 4, and those are the purple ones. And these two mRNAs are translated into different but related muscle proteins. mRNA degradation. The lifespan of mRNA molecules in the cytoplasm is important to determining the pattern of protein synthesis in the cell. And in multicellular eukaryotes, these mRNA typically survive for hours or days or even weeks. And an example can be hemoglobin polypeptides and nucleotide sequences that affect how long an mRNA remains intact are often found in the untranslated region at 3' end of a molecule. And translation is another opportunity for regulating gene expression. This commonly happens at the initiation stage, and the initiations of translation of mRNAs can be blocked by regulatory proteins, which we said are a gene that codes for a protein such as a repressor, and it controls the transcription of another gene or group of genes. And these bind to specific sequences or structures within the untranslated region at the 5' end of mRNA, preventing the attachment of ribosomes. And other blocking mechanisms can be seen in a variety of mRNAs in the eggs of organisms. All right, translation of all mRNAs in a cell can be regulated simultaneously. In a eukaryotic cell, such control involves the activation or inactivation of one or more protein factors that initiate translation. And these play a role in starting translation of mRNAs that are stored in eggs. And it goes on to talk about after fertilization, what happens and how it's translated and triggered by the sudden activation of translation in initiation factors. Are there any questions? The final opportunities for controlling gene expression occur after translation. Eukaryotic polypeptides must be processed to yield functional protein molecules, and this happens with chemical modifications. Regulatory proteins are commonly activated or inactivated by the reversible addition of phosphate groups, and proteins destined for the surface of animal cells acquire sugars. The cell surface proteins and many others must also be transported to target destinations in cells in order to function. And this regulation might occur at any of the steps involved in modifying or transporting a protein. The length of time of a protein functions in the cell is strictly regulated by the means of selective degradation. To mark a particular protein for destruction, the cell commonly attaches molecules to a small protein called ubiquitin to the protein, and gain protein complexes called proteasomes that then recognize the ubiquitin tagged proteins and degrade.